Okay, so if you missed class Monday, October 8th or Tuesday, October 9th, these are the notes you missed in biology. And this is chapter 7. It was chapter 4 in the old book, but not that that really matters. Okay, so we'll skip over the objectives, but by the time we get done, you should kind of be able to say all these. All right, a little history of cell biology. Hooke was the first guy to, sorry about that. Hooke was the first guy to really use a microscope and to examine a piece of cork under the microscope anyway. And he looked at cork because he just had it handy. It was, Captain Weinball or something. And when he looked at the cork under the microscope, he saw these compartments or cells, and so he coined the term cells. I think it's kind of ironic because cork is actually dead tissue, and we use cells to describe living things now. Another guy that's kind of famous in the history of cell biology is Anton van Leeuwenhoek. And he looked at scrapings from his teeth under the microscope, saw some bacteria. He also looked at protozoans under the microscope, which are little unicellular pond swimmies. And this was kind of his first microscope with a little lens there. Not much, but he did see some things. So what is the cell theory? Scientists take Hooke's and Leeuwenhoek's ideas and combine them into a cell theory. And there's 3 scientists that are also credited with it, which you'll learn here in a second. The first statement is that all living organisms are made of one or more cells. We've already discussed this when we talked about the properties of life. Cells are the basic units of structure and function, meaning that everything builds upon a cell in the living organism world. Cells make up tissues, tissues make up organs, organs make up organ systems. So you kind of build upon the cell. And then you already learned number 3 also. Cells only come from the reproduction of existing cells. Remember, that's the theory of biogenesis, that living cells have to come from other living cells. We're going to skip that little video. The scientists that are credited with the cell theory, Schleiden, he was a German botanist. He studied plants, and every time he looked at the plant under the microscope, he said it's made of those cells that Hooke was talking about. Didn't matter whether he was looking at the roots or the leaves or the stems, all parts of the plants are made of cells. Theodor Schwann, he was a zoologist and he studied animals. And he looked at rat liver and lung and that kind of stuff under the microscope, and he said that all animals are composed of cells. And then Virchow, he was a physician. He studied germ theory, the idea that it takes bacteria and viruses to make us sick. And he said that cells only come from existing cells. Again, supporting the work that Pasteur, Spallanzani, and Redi did, which we learned about their experiments. They all proved the theory of biogenesis, and Virchow said that that was true. So these 3 guys are really credited with the cell theory, which you just saw on the other slide. Okay, what are the 2 types of cells? Prokaryote. You might circle pro, means true. These cells, or means before. My bad, it means before. These are cells that do not possess a true nucleus. They came before a nucleus. A nucleus is simply just kind of where the DNA hangs out. It would be down here on this cell. They still have DNA, which is all this stringy stuff. They just don't have a nucleus. A eukaryote cell, by the way, some examples, and I forgot to put them on here. Write down bacteria. Bacteria are prokaryotic cells. They don't have a nucleus. Eukaryotes are cells that possess a true nucleus. That would be like, so write down these examples. Humans, any other animal, plants, those all have true nucleus. Humans, animals, plants. And that means you just have like a compartment that holds your nucleus, or holds your DNA. All right, so cell size. Cells are limited to how big they can become by 2 factors, surface area and volume. Cells have to maintain a large surface area because they exchange stuff with the outside environment. Oxygen comes in, carbon dioxide goes out all the time. So if you have a large surface area, that means more opportunities for things to get in and out of your cell. They have to maintain a small volume because if it's too big, stuff can't get across the cell very easily. Think about if we compare New York City to Abilene. New York's got a lot of volume, a lot of buildings and traffic and people. Takes forever to get across the city. Abilene, not a lot of volume. Doesn't take too long to get across the city. So if you're a cell, you want to be like Abilene, small volume. Doesn't take too long to get across the cell. So small cells are way more efficient, okay? This picture shows kind of a little bit about the lab we did. We cut 3 different size cubes out of this agar. It's kind of like Jell-O, only has chemical in it. We cut a 3 centimeter cube, a 2 centimeter cube, and a 1 centimeter cube. This one has the biggest surface area, but this one has the smallest volume. Well, how are you going to get both of those combined into one cell? So that's the little experiment we did. I will either video the experiment, or I think there's actually a YouTube video that I'll provide you a link for that kind of explains it. There's also another YouTube video that we watched in class about a biology teacher, and it's just a quick 3-4 minute video. I highly suggest if you missed class to watch that because he does a good job of explaining surface area and volume too. So those are the notes we took. The lab, I'm going to provide you a link. I want you to fill out the lab with me. I'll have to make the EduCreations thing. I'm probably going to make that tomorrow. And so tonight, today, if you want to watch the video, fill out the notes, and then watch the couple YouTube links I provide you, and then you'll have to fill out the lab so you understand what we did in the lab. Okay, thanks.